Hello, everyone. Welcome to this podcast from the Light Review. And today we're talking with Professor Glen Jeffrey, Professor of Neuroscience at UCL in London. Bit of a tautology there, University College London in London. The topic is going to be infrared, which seems to be becoming a thing. And what we're going to be talking about, I think, Glenn, if you're happy with the phrase that we've been using the last couple of weeks of mitochondrial infrared, because that seems to be what the infrared is doing that we are talking about. Am I right?
Yeah,
absolutely. Well, excellent. So we can go, well, it's MIR, isn't it? So, how infrared affects the body. It is the new thing. We thought we'd got somewhere with circadian lighting. And then people like Glenn come along, tap you on the shoulder and go, you ain't finished yet, my friend. So I was lucky, along with Shelley James and a few other friends of ours, to sit through this presentation that Glenn is going to give us now. And I think this is so valuable that we're going to put this one up as its own presentation. And then we can all then point at it and the conversation will carry on from there. So Glenn, thanks ever so much for joining us. I'm going to just shut up and just sit here and nod my head at you. And then we'll have a little bit of a chat when we get to the other end. But Glenn, it's over to you, my friend.
Great. Thank you very much, John. So this talk is really about infrared light. But when we use the term infrared, we're spanning near infrared to infrared. So big chunks of it we can't see. And you tend not to talk very much about things in life that you can't see. And we tend not to pay too much attention to what those things do. So that's why part of the story has been, relatively hidden for a long period of time. But let's start with a question. Where do you get the energy from to pull your legs out of bed in the morning? And what has light got to do with it? And I want to talk specifically about sunlight and what elements of sunlight are missing now in our world and what the consequences of that are for us. Right, so let me introduce you to mitochondria. Mitochondria are organelles. There's hundreds of them in the cells in the body, and they are your battery. They provide the energy. Now, a long, long time ago, billions of years ago in evolution, cells, like this character here, didn't have much energy. And the consequence of that was they didn't do very much. And they lived in a world that was very, very simple. And it was mainly formed of slime, wet slime. And evolution progressed. And as evolution progressed, these cells were approached by something called a mitochondria. And the mitochondria said to the cell, here's the deal. You let me live inside you, and I'll provide you with all the energy that you want. And suddenly, life was transformed on Earth. Sunlight drives the mitochondria gets into the cell then takes off and has mobility, and our world changes from a world of simply very immobile slime to a world in which evolution develops. But the thing about the mitochondria is he played a trick. It played a trick. And the trick is, it's a bit Faustian, which is that he got into the cell, he provided the cell with energy, but he didn't tell the cell one critical thing. And that is, I'm coming into your world, but I have the ability to kill you. So you will now have a glorious life, but it won't last very long. And the thing that the mitochondria is very interested in, what drives the mitochondria, the cellular battery that gives you all this extra energy, is it needs sugar, it needs carbohydrate, and it needs oxygen. And ideally, it needs sunlight. So the stranger was bacteria. The mitochondria fundamentally was a bacteria. But I would like you to view her, as just a battery. We can get through some really big concepts if we just talk about batteries. And these batteries drive our cells. They do lots of complicated things. The harder you work the battery, though, think about your car, and the older you get, the battery runs down. So you have problems swinging your legs out of bed first thing in the morning as you get older because you've got less battery power. And that battery power, it produces something called adenosine triphosphate, ATP. ATP is the liquid energy that the battery is producing. And this fits into what we call the mitochondrial theory of aging. That mitochondria came in, it was a battery, but it meant that you were no longer relatively immortal. You were mortal. And this gave rise to the mitochondrial theory of aging. And that's really one of the main theories of aging that we have at the moment. And what it says is that at the end of the day, the battery runs down. And when the battery runs down, you die. So it's really important that we find out what runs batteries and who needs batteries. Well, all of your tissues are not the same. Some parts of your body need more energy than others. And surprisingly, the part of your body that burns more energy than any other part is your retina. Your retina consumes vast amounts of energy. In fact, you produce your own body weight in ATP each day. It's produced, it's degraded, it's reproduced. The other part of your body that uses vast amounts of ATP is your brain. Your toes don't really need very much. And because your brain needs vast amounts of energy, when the energy runs down, we end up in situations where we have Alzheimer's disease, where we have Parkinson's disease. In the retina, we end up with macular degeneration. They're all linked to the things that our mitochondria are doing to us because they've imposed mortality on us. Okay, so what's the relationship between these mitochondria and light? They like light. Let's slip over and talk a little bit about light, then plug that in to mitochondria. So life has evolved for billions of years under sunlight. And what we have on this slide here is the spectrum of sunlight. It goes from across our visual range. It extends right up to 250 nanometers. 2,500 nanometers. We can't see that. That's all out here. But that's influencing our body. And it also extends down to the ultraviolet. What I'd like to make clear, though, and what I want you to think about, is different wavelengths of light penetrate the body to different extents. So your ultraviolet light doesn't go very far, and that's why you get sunburn, because the skin is blocking it and the energy is being released superficially in your skin. But these super long wavelengths here penetrate very deeply through the body. You can get 800, 900 nanometers. You can measure it coming out the other side of your body. And as it passes through, it provides energy for reactions in the body. So light, there's a fundamental relationship between light, your body, disease, and your life expectancy. So let's break those wavelength components up. Mitochondria, you can imagine them looking up at the sky. They're looking up there and they're testing what wavelengths are in the sky. Now, if we look at those deep reds, the 650, the 900s, the deep reds that extend into the range, we just can't see anymore. They're absorbed by the battery, by the mitochondria. And what it does, it recharges the battery. And that is literally what it does. The mitochondria have got a little charge on them between the inside and the outside, and that charge gets bigger as they absorb longer wavelengths. They produce more ATP. You've got more energy to kick your legs out of bed in the morning. They can be used positively in models of disease. Lots of animal models have shown these longer wavelengths are particularly positive in dementia, in aging, because of the mitochondrial theory of aging. But the other wavelengths of light, let's look at the other end of the spectrum, which we can see relatively clearly, 420 to 450 nanometers, which is in the blue, deep blue, working its way into the violet. That discharges the battery. We know exactly, we've got a pretty good idea how those mechanisms work, but it discharges the battery. It reduces the ATP and it can increase the pace of aging and increase the probability of death. But remember, in ordinary sunlight, which we've evolved under for millions of years, there's a rough balance between the blue and the red. So one of the questions we've got to bear in mind is, what happens if we kick that balance? What happens if that balance changes? What does that mean for us? So here's a simple experiment. And this experiment is where we give 670 nanometer light, which we know increases ATP. And here we have some populations of mice, and we've exposed those mice to deep red light. They're old mice, they're aged, and here is their retina. This is a control animal. Nothing's been done to it. And this animal has had a burst of 670 nanometer light. It's got more ATP. Let's move over to the top of the brain, the cerebral cortex. The animal that's had nothing done to it, how much ATP it's got. And this one here has had 670, and it's almost got twice as much ATP in it. The brain is a three-dimensional structure. It's very deep, but remember, long wavelengths go deep. And this is deep in the brain, called the thalamus. Again, the control, we did nothing to it, measured the ATP. And here we've measured it in the thalamus. And again, in both superficial and deep regions of the brain, we've got an increase in ATP. This is a complicated slide. I don't want you to pay too much attention to this slide. I want you to follow immediately kind of just what I'm pointing at. So what we did here is we put a little electrode on the mouse eye, and we can measure what the retina is saying to the brain. And it comes in series of waves. What I want you to pay attention to is the size of the waves. A young mouse, 2-month-old mouse, look at the size of the waves. That's how the retina is shouting to the brain, passing messages on. Here's a 7-month-old mouse. It's got old. Mitochondrial theory of aging. Those waves aren't as big anymore. But here's that 7-month mouse a little later on in the day, after we've given it a burst of 670 nanometers, and the waves have gone up. So you can think about, if the retina is talking to the brain, it has a reduced ability to do that with age, but we improve it when we give it 670 nanometers, and we improve its mitochondria. Here's another one. I don't want you to look at this. I want you to just squint when you're looking at this. Your foot ages at the same rate as your brain. Okay, they're under slightly different pressures, but you can't have your foot aging at twice the pace that your brain or your liver are aging. It has to be a harmony. So when we shine red light onto an organism, there has to be a unitary response to that light. And what we've looked at is we looked at the cytokines that are running around in your blood. They're inflammatory markers. They're messengers that tell different tissues in your body what's going on. Just if you look at the patterns here, the black lines are the cytokines in control mice. Nothing's been done to them. The red histograms are cytokines in the bloods of mice that have been exposed to red light. There's only one point that you need to take from this, and that is, if you shine red light on one part of your body, other parts of your body get to hear about it very soon. That is really quite important. It's quite important for how we use light. I used to have the fantasy that we could give people red light by putting their hand into when they go to the cash machine. While they're at the cash machine plugging in their numbers, they put their other hand down and it gets exposed to red light. And I said to everybody in the lab, this is a fantasy, but we're going to drive towards that fantasy. That's not a fantasy anymore. We now know that we can shine red light on part of the body, and the mitochondria around the rest of the body all respond to it. They talk to one another. So, big pictures. The mitochondrial theory of aging. If we improve our mitochondria, we improve our health span, and according to the mitochondrial theory of aging, we might live longer. Well, the only realistic way we can do this is to use a very, very simple animal. And so we use the laboratory fly, the fruit fly, and we take 200 flies. And we count how many flies we've got in each batch of 100. And in one of those batches, we don't do anything. In the other batch, we give them a burst of red light every day. And on this graph, these are the weeks. A fruit fly lives about 12 weeks. The dark line are the control animals. You can see how many they've got. So by the time we get to about 6 weeks, about half of them are dead. And by the time we get to 12 weeks, they're all dead. But the flies that are exposed to red light each day are dying at a much, much slower rate. So by the time we get to 9, 10 weeks, we've got twice as many alive in the red group than we do in the control group. Now, you're not living longer, but what's happening is those middle-aged deaths that are occurring in control animals are occurring at a much lower rate in animals that are exposed to red light. Now, that fits perfectly with the mitochondrial theory of aging. We're improving our mitochondria, they're producing more energy, the flies are living longer. And if we measure how quickly they can climb up a tube, we find that this improvement in lifespan, quality of life, is reflected by their ability to climb. They can climb almost at twice the rate that control flies can climb. And one point I really want to make here that's important is everything we find in a fly, we find in a mouse. Everything we found in a mouse, we find in us. So here's a sort of a school kid type experiment. You take a group of flies and you cut their wings off. That's the first thing you do. And you leave them for a week. Then you plug them. Now you're looking down at 3 islands. And the islands have got moats around them. Water. So the fly can't fly. He's running around. And what a young fly does is he runs round the edge of the moat. And he's trying to find a way out. Older flies, less ATP, mitochondria run down. They get lost. They simply get lost. They don't have a strategy for getting out. If you give these mice a burst of red light, you don't take them back to where they were, but you improve their strategies for exploring their environment. We don't have all these squiggles in the middle in these guys here. We've not taken them back to youth, but we've eroded some of the issues in aging. And this is in our cognition. It's not motor skill, it's cognition. A terrible graph again, forgive me. But I only want you to look at 2 things. So we have a young fly, we have an old fly. We have an old fly that's been given red light. So locomotor function, we discussed that. Old fly, young fly, old fly goes down. Old fly with red light, it improves. But here's a real good cognitive one, because we're asking memory. We put a fly in a tube with a light at the end, and the first thing the fly does is run straight towards the light. Flies love light. Well, then we do something horrible, which is we put a piece of filter paper in the tube with quinine in it. Flies hate quinine. They loathe it. And if you get the fly to run down that tube, that fly will remember it's quinine and he won't go towards the light with anywhere near as much enthusiasm. So young flies remember that there's quinine there and that affects their performance. 6-week-old flies, they're starting to forget. They're running down the tube towards the light and they're starting to forget. Take these old flies and you give them a burst of red light. You improve the mitochondria in their brains, in their mobility, and lo and behold, they behave like young flies. They're not going to run towards. They remember there's something nasty down that tube. So that's really important. We're not only improving your health span, your ability to get out of bed, but we are in these animals improving their ability to, think, remember, cognitive powers. So when we did this, there was quite a lot of, black magic, including from John, actually, I should say, in the early days. And I'm looking in the world for a mechanism. I'm looking for something which adds weight to what is going on. And it occurred to me, I always tell this story because it was a dramatic event. I was cycling up Highgate Hill. I had a wind against me and it was pouring with rain. And I had this Damascus moment, this sudden flash. And that is the insecticides we use to kill insects that we're spraying. The neonicotinoids, which are partially banned in the UK, have a disastrous effect on insects, including bees, and they develop what is like a Parkinson state. They discharge the battery, there's no ATP, the animal can't move, the animal doesn't feed, the animal dies. So what if we poison the animals? And what if you gave them red light? Now that would clearly show that what we're doing is we're working on mitochondria and we're having an effect. So the first thing that we did was we took the bees and we poisoned them and we looked at their mobility. And here are those guys, we poisoned them, their mobility really declines. But then we also have our controls here in blue that weren't poisoned. But then we have the guys that we poison. We poison them with something called imacloprid. You'll pick it up. It's a hot topic for discussion, the use of imacloprid. And the point is, the first kind of day afterwards, they're a bit hungover. They're not universally happy. Their mobility goes down a bit. But as time goes on, they're protected. They're protected from this mitochondrial damage. And we can say the same about their lifespan. If we look at ordinary animals, our control animals, which are blue line here, our blue line, most of the bees, these are worker bees, most of them are dead after 31 days. The guys that we give imacloprid to, where we damage their mitochondria, they're in a poor state and they're dead at 20 days. But like the fly, what we have here is these are the controls that just had the red light. The red light, they live longer. All those middle class deaths, they're going. The imacloprid and the 670 here are almost the same as the control. So we're affecting cognition, we're affecting mobility, and we're affecting lifespan, which is good news for us because we're an aging population. And this has really taken off in certain parts of the world, particularly in the United States, where they've used insecticides so heavily, they've destroyed all the pollinators. And this is true in California, and particularly for the almond harvest. And so what the Americans do is they truck up thousands of bees from Texas and Florida on these lorries. They let them out in certain areas. The animals pollinate the plants, and then they drive them back down to Texas or Florida. But the price they pay for that, this is a terribly stressful event for a bee, terribly stressful. Opens a door every day and it's in a different place. They really don't like that. There's a 40% loss of hives in these lorries, in these trucks. So we've now got red lights in these hives, in these trucks, and loss of hive is down by about 70%, which keeps the accountants happy who are running these companies. They don't particularly care about bees, they care about money. So let's now take all this together with us. And this is the important thing. At the end of the day, if we're going to do experiments on animal models, we've got to translate it. Now, we're not doing anything like drug tests or anything complicated. We're just giving people bursts of red light. So we have groups of people here, and this is their age span. Let's just look at this top graph here. Now, this is one person. Every pair of black and red is one individual. And what's measured is your ability to see a coloured letter in noise. We want to find out where is your threshold to see that letter and identify it. And you can see what the letters are like here, rather poor. So it's a threshold. That means going down is good. So we do a baseline measurement on all these people, which is the black dots. We give them a burst of red light and we retest them 3 hours later. And for each pair, the red square underneath is the threshold for that individual. And you can see that in each case, the red square is lower than the black dot. That means thresholds have gone down. That means vision has improved. And that's for a blue target. This is for a red target. And if we look at the percentage threshold improvement for blue, blue's always Blue is a difficult issue for the retina. Your blue system is very, very vulnerable. But we managed to reduce your threshold by 17%. That's quite a bite. For the reds, we only managed to reduce the threshold by 12%. But we've improved the ability to detect colours on a background of noise. Translational research. How long does this kind of thing last, a burst of red light? Well, it's the same whether a fly, a bee, a mouse. It's exactly the same. It lasts just under a week. I'll give you a 3-minute burst of light. So these groups of people here, they've had a 3-minute burst of light, and we've retested them 1 week later, and you can see the red is still below the black in many cases, but not all. These people have returned to where they were before. These people have returned to where they were before. But some of these are still showing improvement. I don't know why some people hold out a bit longer and others don't. Just beyond me. Same for the red. The effect lasts approximately 1 week. So what else can we do with red light? And this is something that I suppose by the time people watch this podcast, this will be published. It has a press release for Wednesday because it's a relatively dramatic effect. Because mitochondria need sugar and they need oxygen. And those are the two things that they need to keep you going. They burn sugar, they burn oxygen, and they produce CO2. So what happens if we give people a burst of red light? We will then increase the demand from glucose, carbohydrates from your blood. They will need that. It will increase the demand for oxygen to produce CO2. Can we use that to reduce your blood sugar levels? Aging population, a relatively immobile population, a population where diabetes is a real major threat. Once you've got diabetes, you've got a whole host of other issues. Potential to go blind, you get diabetic ulcers, there's a chance of having your legs amputated because your blood system collapses. So, horrible experiment that we had to drag our friends in to do. We got them to drink a large amount of glucose, standard glucose tolerance test. Then we pricked their finger to measure their blood glucose, and we measured the amount of CO2 they produced. The green line here are our control group. So approximately 40 to 60 minutes after they've consumed this horrible glucose solution, their blood glucose levels peaks. Peaks in blood glucose are the really bad thing, going up and coming down. If it's up and stays up, actually that's not really that harmful. These are the people that had a burst of red light on their backs. Their blood glucose levels do not go up to the same amount. And if we measure this by area under the curve, their situation is a lot better, a hell of a lot better. And remember, this is a burst of light on your back. Remember, mitochondria talk to one another across the body. So that mitochondria on your back gets the burst of red light. It increases its metabolism. It increases its rate of respiration. But then it tells all its friends in your liver, in your kidneys, and in your feet to do the same. So we have quite a dramatic effect on blood glucose. To prove that this is the mechanism we think it is, then this should be associated with an increase in oxygen consumption, which would mean you produce more CO2. And lo and behold, the people that have had the red light produce more CO2. Now, the point that I'm heading towards here, and I think it's particularly an interesting point for the lighting industry, and those interested in health in the lighting industry, is I've told you about the things that we can do with red light. I know what we can do with blue light, but I'm not going to do it because I wouldn't get ethical permission to do it in humans to start pumping up their blood glucose. But what is it about the light in our environment that could influence this? So again, if we go to this figure over here, this is the light spectrum in sunlight, roughly. We've got our visible part. We've got a big chunk of infrared, a very big chunk of infrared. This light, as many of you will recognize, is basically the light from an LED. We've got a peak in the blue, which I'm personally very unhappy about. But also we have very little red. Our red's truncating just over sort of 700-ish. In natural sunlight, we go all the way to over 2000 nanometers. We've got something missing here, and that thing that's missing, I've just shown you, plays a very important part in biology, regulating aging, cognition, and health. This is one of the old eco bulbs, which I think now are rather largely out of favour, but again, there is no red light. Now, if we were to go back to 30 years, we would have incandescent light bulbs, filament bulbs. Those filament bulbs are very, very similar to natural sunlight. They've got a big chunk of red in them, an infrared. So up until the mid-1990s, we were actually okay when we lived inside, because as we lived inside, we had incandescent light bulbs with a load of infrared in them. We're now here. That red's gone. Just as a little sideline, which I regularly have a dig at, this is the spectrum of some of the new BMW laser lights. Apparently they're absolutely stunning. You can see for miles in the darkness with them. But I think it was the Sunday Telegraph or the Daily Telegraph motoring correspondent said, if you get caught in the beam of one of these things, it's not about you're okay 3 minutes later. There is something going on. You bet there's something going on. Look at that peak in the blue and think of your mitochondria. Your mitochondria crying in that one. Blue. Let me just run you why your mitochondria are crying in blue. This is an experiment where people have been cannulated, so we can measure everything that's going on in them, their blood pressure, really easy. So what we do is we then give them, I certainly did not do this, we then give them a burst of 450 nanometer light to their body. So, and we measure a number of metrics. Heart rate shoots up when you're exposed to blue light. Blood pressure nosedives when you're exposed to blue light. Those are major changes in your body that are arising as a consequence of 450 nanometer light. Now, the long-term consequences of that have never been thought about, but I'm very unhappy about moving into an environment where those sorts of things are happening to me. And I really don't know why this very nice study, which partly came out of the University of Surrey, has not been discussed more widely. This is data that isn't published, but I think it's absolutely great. This is done by one of my ex-graduate students who is Dean of the medical school in Kuwait. And she asked me, well, you know, what else goes on with blue? And I said, I can't do it. I'm not doing experiments on blue light on people. You know, it's just not ethically sound. So she took a whole group of mice and she exposed them every day to blue light. Now, blue light slows down your mitochondria, influences your metabolism. 450, loads of 450 in our environment. These are the control mice and these are their weights. The controls stay relatively stable. These are the mice that are exposed to blue light. We've slowed down their metabolism. We've slowed down their mitochondria. And because we've slowed down their mitochondria, the mitochondria are not drawing sugars out of our blood. So the sugars are running round. And in the end, body's saying, let's take them into storage. Let's save this for a rainy day. Consequence is, you put on a very large amount of weight. These are weeks. These animals are 3 times heavier at this stage than the controls. They look awful. They look absolutely awful. But it fits. It's the mixture between blue and red. In an environment where blue and red are mixed, mitochondria are in an equilibrium. So the argument that I want to make is that what we're dealing with here is a significant issue in public health. We are being starved in the built environment of red light. At night, when you go out, many of the street lights are very rich in blue, and there's very, very little red in them. Particularly true of things like sports stadiums, very intense, hard white lights. So we've got red light starvation in the built-in environment. We've got an aging population that is red light starved, and we know that that aging population in a red light starved environment is likely to be more vulnerable to degenerative diseases that are regulated by mitochondria and to diabetes. Reducing mitochondrial performance and reducing ATP will drive aging. Mitochondrial theory of aging comes out of the late 1950s, but it's solid. Every time you look at it, you think, well, that fits. That fits. It's not a fad. That is a really solid theory. So we need to think about how we supplement our built environment with red light, or much more eco-friendly, we need to walk outside. We need to spend more time in sunlight. And an interesting angle on that one is when your plant outside is sitting in very bright sunlight in June and you touch it, it's not hot. It's not hot because it won't absorb infrared light, it reflects it. So you can get a very significant exposure to infrared light by walking in the park. Makes me sound like a tree hugger. I'm not a tree hugger, but I think that's an important point. Okay, that's my story. And I would like people to go away with the idea that we're talking about public health. We're talking about it within a framework of an aging population that is vulnerable and will be costing us a lot of money. That's my story.
Glenn, and that's quite a story to be telling, and thanks ever so much for all of that. It raises all kinds of issues and all kinds of questions, and I don't really want to be here for the next couple of hours so just going through all this, because we will find other platforms to do that, I'm sure. Just one thing though that I just like to ask you: the blue pump on the LED, that huge peak that we've got. Generally speaking, in commercial LED production, we don't have a choice on that. That's what makes the LED work.
Yes.
Yeah. Now, if we and what you and I have been talking about is bringing in infrared in some way into our environment. Now, how does that work with the balance? The blue is still there doing its bad stuff, and the red is there to put back into our light diet, as it were, good things. But is there then a battle royal going on inside us, because we've got blue light wanting to do one thing to us and the red light wanting to do something else to us? Or is there some kind of melding that goes on? Do we know?
I think it's a very good question. So the big thing everybody wants to know, how much? How much blue, how much red?
I think I've asked you that one. Yeah.
Yeah, exactly. And it's a natural question to ask. Part of the answer resides in the fact there's enormous amount of slop in the system. Okay, so you can have, as long as you are very roughly in balance between the blue and the red, it's fine. Now, we do things that are pretty kind of horrendous in the lab when you think about it in terms of metabolism. We give a hefty chunk of, say, 850 nanometers for 3 minutes. But in actual fact, we can do that for 20 minutes. It makes no difference, right? I mean, there is a lot of slop in the system. This is not like taking paracetamol when you've got a hangover. I'll take 5 paracetamol because I'll get over this hangover quicker than if I take 1. It doesn't work like that, right? And that's really important, because most of the medics think of anything in terms of a dose response curve. You take more, you get better at a faster rate until you fall off a cliff at the end. It just doesn't work like that. The mechanism is much more switch-like. You get enough red, and that is enough to get you through the situation. So there's a lot of slop in it. If you give less than 1 minute's red light, nothing happens. If you give 3 minutes, it's great. If you give 20 minutes, that's as great as the 3 minutes. And now I don't understand that. It really, really bugged me for a long time. But there comes a point where you've got to say, well, look, you know, I don't have the mechanism at the moment really to understand that. Let's push on a little bit by making this something that's addressable in terms of public health.
Okay. Now, all right. And then one last question, and then we'll say thank you very much. When we look at the breadth, the bandwidth, if you like, of the infrared, and we realise there's more of it going on than there is in than the visible spectrum across the whole of that. And then we talk about 850 nanometers. A monochromatic. I know it's not chromatic. A monochromatic response, and then we get a lovely peak. But does that work? Does that lovely low intensity across the infrared zone is that balanced by one spiky peak at 850, or are we losing something in that process?
I think you're right. I think I'm very wary of the fact that are we losing something, right? And because sunlight, which I'm holding up as being the most important thing because we've evolved under sunlight, giving us one particular spike is not sunlight, right?
No,
I mean,
yeah.
I regard sunlight as the cure of the disease. I regard 850 nanometers as being a very good sticking plaster over the problem. So sunlight is the answer. If we can't get it, then we should be thinking about broad spectrum infrared lights. Now, when you look at it in sunlight and you think, well, you go out to 800, 900 nanometers, at any one point, there isn't a lot of it there. However, the key issue is those wavelengths penetrate deep in the body. The longer the wavelength, the greater the penetration into the body, and hence the greater of the ability for it to affect physiology. Right? So I cannot get 670 nanometers, which has been a wavelength very commonly used. I can't get it through my chest and out the other side. 800 plus, I can measure it coming out the other side. So more tissue is exposed to those wavelengths because they penetrate more deeply. So there's a number of different issues we've got to take into the equation. Now, you could have 53 PhD students working on this one for 20 years. And that will be the measure of the problem, because you'd have to go from wavelength to wavelength to wavelength and say, how far is it going? What energy is it dropping in what tissues? Because different tissues have got different optical penetrance.
Yeah.
So, again, I'm going to go back to the public health issue. I'm going to say, if I can get rid of the blue light and give you a bit of red, I'll die happily.
Okay, so looking at it then as a lighting chap, we know that the standard commercial LED spectrum, which has got that huge, great blue peak, is not necessarily the answer, because we see full spectrum LEDs out there that do not have that peak. So we assume from what you're saying that if we can shift to that kind of LED quality, then we're probably helping ourselves.
Yeah, definitely. I think and I've seen a number of products that have come out from the Far East which call themselves sunlight. They don't have that harsh peak.
Yeah.
And I should point out that harsh peak hardly changes between warm and cold LEDs. But what they have not got is they have not developed the technology to extend the emittance all the way out to places like 900 nanometers. That is what we need to do. Now, you're then, when it comes to the lighting industry, they're saying, well, why am I doing this? Because you can't see it. So there has to be this point where the mitochondrial light people meet the lighting engineers, and it's quite difficult because the languages are quite different, and some of the concepts are quite challenging for both of us. And we're asking you to generate light that we don't see, that's costing money. I get it. Yeah. There's an issue there.
Yeah. Okay. Right. And at that point, there's an issue there. There certainly is. Let me just say to everybody who's watching, thank you very much for your time. I hope that has what should we say, has lit a bit of an infrared fire under us to see where we take this one, because clearly this is a health issue. This is not an illumination issue. There are people out there, and I've been one of them, saying, why are we talking about this? But I think we have to talk about this because this is our toolbox. These are our spanners and screwdrivers that will get us into or out of this. So, Glenn, thank you very much for your time. I'll be seeing you again very, very soon. And as I say, this topic is going to run and run. Thank you very much.
Thank you. And you guys in the lighting industry are super important. Really super important for public health. So thank you for giving me a chance to stand on my orange box.
Excellent. Thanks, Glenn. Cheers.
Take care. Bye.